Welcome, everyone. I'm Ralph Clayton, host of the VTM Podcast. This is episode 23. Today, I want to take you into one of the strangest and most promising frontiers in modern medicine. Medical microrobots, nanorobots, targeted drug delivery, bubble-based movement, lung treatment, nanobiosensors, liquid biopsy, carbon nanotubes, machine learning, and the possibility that future medicine may not only flood the body with drugs, but send tiny engineered systems toward the place where help is needed most. This is not simply a story about miniature machines. It is not simply a story about cancer detection. And it is not only a story about technology becoming smaller and smaller until it enters the bloodstream. At its deepest level, this episode asks one dangerous question. What happens when medicine learns to move, search, sense, and act at the scale of disease itself? Because disease is often local before it is visible. A tumor begins with cells. A clot begins with a hidden obstruction. An infection begins in tissue. Inflammation begins in small signals. A damaged vessel changes chemistry before the larger organ fails. A brain tumor may alter the blood long before diagnosis is simple. A lung infection may live in spaces that ordinary drugs reach poorly. A cancer may hide behind barriers that protect the body, but also protect the disease. Modern medicine has become powerful, but much of it still operates with a blunt logic. A drug enters the body. It circulates. It diffuses. It reaches many tissues. Some of it reaches the target. Some of it does not. Some of it helps. Some of it harms. Chemotherapy can attack cancer, but also injure healthy tissue. Antibiotics can treat infection, but also spread through the system. Anti-inflammatory drugs can reduce disease activity, but affect the whole body. Even advanced medicine often faces the same old problem. How do you get the right therapy to the right place? At the right dose, at the right time, without damaging everything else on the way. That is the first great promise of medical micro and nanorobotics. Targeting, not magic. Not science, fiction swarms thinking for themselves. Not tiny humanoid robots walking through arteries with little tools. The real field is stranger and more practical than that. It is about engineered particles, microscale swimmers, magnetic capsules, biohybrid carriers, ultrasound-responsive bubbles, algae-based micromachines, hydrogel spheres, bacteria-inspired motors, and nanosensors that change their optical signals when molecules bind to them. It is about motion, sensing, steeraging, cargo, release, biocompatibility, clearance, and control. The fantasy version imagines nanobots as miniature doctors. The real version is closer to programmable matter in biological environments. Tiny systems designed to survive inside fluids. move through complex spaces, carry drugs, respond to signals, become visible to imaging, release payloads, or detect disease signatures that ordinary tests may miss. That is still extraordinary. But it is not yet the science fiction version. And that distinction matters, because this field sits near the boundary between wonder and hype. The word nanobot can mislead people. It can summon images of self, replicating machines, hidden swarms, or microscopic artificial intelligence inside the body. That is not what current medical research is delivering. Today's medical micro and nanorobots are usually simple in their individual behavior. They may move under a magnetic field. They may be propelled by ultrasound. They may use bubbles. They may use enzymes. They may use the natural motion of a microorganism. They may respond to chemical gradients. They may be guided by external imaging. They may carry particles or drugs. But they are not miniature, conscious robots. They are engineered therapeutic or diagnostic systems. The intelligence is mostly outside them. In the design, in the material, in the field that guides them, in the imaging that tracks them, in the algorithm that interprets sensor signals, in the clinician who decides where they should go. That is the second lesson. The future of robotic medicine may not be tiny robots thinking inside us. It may be tiny bodies obeying carefully designed physical rules. And sometimes that is enough. Consider targeted drug delivery. A drug that treats cancer may be powerful, but power spread everywhere becomes toxicity. If a medicine could be concentrated more precisely at the disease site, the same therapeutic effect might be achieved with less systemic harm. Or a stronger local effect might be possible without increasing whole body exposure. This is the ancient dream of targeted therapy. Antibodies do part of this. Nanoparticles do part of this. Liposomes do part of this. Implants and local injections do part of this. But microrobots add another idea: movement. The carrier does not merely drift. It can be guided, or propelled, or stimulated, or made visible. It can become part drug carrier, part contrast agent, part delivery vehicle, and part controlled release system. That is why magnetic microbots are so important. Magnetism can act through tissue. A magnetic field can guide a responsive object without wires. This makes magnetic control attractive for navigating tiny devices inside fluids, vessels, cavities, or organs. But the body is not a simple tube. Blood flows, mucus resists, immune cells attack, tissues deform, organs move, fluid environments differ radically. The stomach is not the bladder. The lung is not the bloodstream. The eye is not the brain. A system that works in a microfluidic channel may fail inside living tissue. A robot that moves in water may stall in mucus. A particle that survives in a dish may be cleared by immune cells. A device that can be guided in a shallow organ may be difficult to track deep in the body. That is why real medical microrobotics is not just about making something small. It is about making something small that can function inside the violence of biology. This is where the field becomes difficult. A microrobot must often solve many problems at once. It must carry a useful payload. It must move or be guided. It must be visible. It must not trigger unacceptable toxicity. It must release its cargo in the correct location. It must avoid uncontrolled accumulation. It must clear from the body, degrade safely, or be removed. It must be manufacturable. It must be sterilizable. It must satisfy regulators. It must work better than a simpler therapy. That last point is crucial. A microbot is not valuable because it is futuristic. It is valuable only if it solves a medical problem better than existing tools. The patient does not need novelty. The patient needs benefit, less toxicity, better targeting, earlier detection, more precise intervention, a disease site reached that could not otherwise be reached, a therapy made possible that would otherwise fail. This is why the most serious work in the field is moving toward practical demonstrations. For example, researchers have tested bioresorbable acoustic hydrogel microrobots for targeted drug delivery. These are not little metal machines. They are tiny bubble-like hydrogel structures. They can incorporate magnetic nanoparticles. They can carry therapeutic cargo. They can be tracked through imaging. They can move using acoustic effects related to trapped bubbles. And in animal studies, they have been used to deliver therapy to bladder tumors. The important idea is not that the robot looks impressive. The important idea is integration, movement, imaging, drug loading, release, and eventual absorption or degradation. The body cannot be treated like an empty laboratory channel. A delivery system must enter, act, and leave no unacceptable burden behind. That is why bioresorbability matters. A tiny machine that remains in the body forever creates a different problem. Where does it go? Does it accumulate? Does it inflame tissue? Does it degrade into safe products? Does it interfere with future imaging? Does it trigger immune response? Does it cross barriers it should not cross? A good medical robot must not only arrive, it must have an ending. This is a strange but essential point. In medicine, the exit strategy is part of the therapy. Then there are bubble-based robots. Bubbles may sound fragile, almost childish. But in medical physics, bubbles are powerful. Microbubbles can interact with ultrasound. They can serve as imaging contrast agents. They can oscillate. They can collapse. They can create local mechanical forces. They can help open barriers or enhance drug penetration under controlled conditions. A bubble is not just empty space. It is a responsive structure. Some new microbubble. Robots use internal bubbles for ultrasound visibility and movement. Some use enzymes to generate motion. Some use magnetic particles for steering. Some use ultrasound to trigger release or collapse. The result is a platform that combines motion, imaging, targeting, and mechanical penetration. That is why bubble-based movement is so interesting. The same feature that makes a robot visible may also help move it. The same feature that helps track it may also help trigger therapy. In the best designs, every component does more than one job. That is a mark of maturity in this field. A microrobot cannot afford unnecessary complexity. At this scale, every part must earn its place. The shell, the cargo, the magnetic material, the enzyme, the bubble, the coating, the imaging signal, the release mechanism. The more functions are integrated into a safe and controllable platform, the more clinically plausible the system becomes. But plausible is not the same as proven. Most of these systems remain preclinical. Animal models, phantom models, microfluidic systems, early demonstrations. That does not make them unimportant. It means we must not mistake the road for the destination. A mouse tumor is not a human cancer clinic. A lung infection model is not every human lung disease. A bladder experiment is not regulatory approval. A beautiful video of particles moving under a magnet is not a therapy. The work is real. The promise is real. But the translation is hard. And translation is where biomedical dreams often fail. Not because the science was fake, because the body was harder than the experiment, because manufacturing was difficult, because long-term safety was unclear, because tracking was insufficient, because the advantage over standard treatment was not strong enough, because cost was too high, because regulators needed more evidence, because clinicians needed a simpler workflow, because what worked once in a laboratory did not work reliably across patients. This is the third lesson. In medicine, a technology is not finished when it works. It is finished when it works safely, repeatedly, at clinical scale, in real patients, under real constraints. That is the standard and nanorobots must meet. Now consider the lung. The lung is an extraordinary target. It is accessible through inhalation. It has enormous surface area. It is exposed to air. It is vulnerable to infection, inflammation, fibrosis, cancer, and environmental injury. But it is also difficult. The lung has branching airways, mucus, surfactant, immune surveillance, macrophages, ciliary clearance, delicate alveoli, and constant motion from breathing. Traditional inhaled therapies already exploit the lungs' accessibility. Asthma inhalers, nebulized drugs, inhaled antibiotics, aerosols. But delivery into the lower respiratory tract is still challenging. Particles must be the right size. They must reach the right region. They must avoid being exhaled, swallowed, trapped, or cleared too quickly. This is where inhalable microrobots become interesting. Researchers have tested biohybrid microrobots based on tiny algae as active drug carriers for lung treatment. The idea is almost startling. Use a living or biologically derived microswimmer as an actuator. Attach therapeutic nanoparticles. Deliver through inhalation. Let motility help distribute the therapeutic system through lung tissue. Use the organism like motion to resist rapid clearance. In one recent study, a nebulizer was used to deliver algae-based microrobots into mouse lungs for treatment of acute MS, pneumonia. The microrobots retained motility after nebulization and carried vancomycin, loaded nanoparticles. The promise is direct, active, pulmonary drug delivery. The caution is equally direct. This is still experimental. The lung is sensitive. Any inhaled material must be evaluated intensely. Immune reactions matter. Retention matters. Clearance matters. Inflammation matters. Dose matters. Manufacturing matters. Still, the idea points toward a future where inhaled therapies are not passive aerosols alone. They may become active delivery systems, systems that move, persist long enough, carry drugs locally, and reduce systemic exposure. That would be a major step, especially for infections that are hard to treat, cancers that need local therapy, or diseases where systemic dosing creates too much harm. This is what precision medicine means at the physical level. Not only sequencing a patient's genome, not only choosing a drug based on biomarkers, but placing the therapy where it is needed, in the right tissue, at the right time, at the right concentration. Precision medicine is not complete if the medicine still travels blindly. Micro and nanorobots may become one answer to that problem. But the other side of precision is detection. That brings us to nano, biosensors, and liquid biopsy. A biopsy traditionally means taking tissue. A tumor is sampled. A needle enters. A surgeon cuts. A pathologist examines cells. The tissue gives truth, but tissue biopsy can be invasive, risky, painful, difficult to repeat, and in some cases, dangerous. A brain tumor is not a skin lesion. Sampling it is not simple. Liquid biopsy offers another possibility. Instead of cutting tissue directly, analyze fluids. Blood, urine, saliva, cerebrospinal fluid. Look for signals shed by disease. Circulating tumor DNA, methylation patterns, proteins, extracellular vesicles, metabolites, immune signatures. fragments of the tumor's existence moving through the body. The dream is a blood test that can detect disease earlier, monitor response, track recurrence, and reduce the need for invasive procedures. But brain tumors have been especially difficult. The brain is protected by barriers. Tumor-derived DNA may be scarce in blood. Signals may be faint. Imaging can show a mass, but not always precisely what it is. Biopsy remains invasive. This is why a 2026 Nature nanotechnology study attracted attention. Researchers reported a machine perception liquid biopsy approach using quantum well defect, modified single-walled carbon nanotube nanosensors, stabilized with stranded A, single-stranded DNA. That phrase is technical, but the basic idea is elegant. Instead of looking for one known biomarker, use an array of nanosensors. Expose them to blood plasma. Let molecules in the plasma form interactions with the nanosensor surfaces. Measure optical responses in near-infrared fluorescence. Then use machine learning to detect patterns. Not one molecule, a fingerprint, a disease signature in the way the sensor array responds. In the reported study, researchers used hundreds of plasma samples from patients with brain tumors and non-tumor controls. The system detected intracranial tumors with high accuracy in the study and also attempted to identify tumor type. It did not solve everything. Tumor type classification was more modest than tumor detection. Some clinically important distinctions remain difficult. The external validation performance was lower than the internal test performance, as one would expect in a serious translational setting. But the deeper importance is the method. The sensor is not only a detector. It can also help reveal what proteins or factors are driving the signal. The nanosensor surface collects a protein corona. That corona can be analyzed. And from that analysis, researchers can identify disease, associated factors from tumor cells, the tumor microenvironment, and the patient's immune response. This is a powerful conceptual shift. The disease is not only detected by what the tumor itself sheds. It may be detected by the wider ecosystem the tumor creates. Tumor, immune system, microenvironment, inflammation, secreted factors, systemic response, disease as a pattern, not one note, a chord. That is why machine learning belongs in this story. Not because artificial intelligence magically diagnoses disease, but because nanosensor arrays can produce complex, multidimensional signals. Human eyes may not see the pattern. An algorithm may, but this also creates a new responsibility. If machine learning helps make a medical diagnosis, the model must be validated. The data must be diverse. The performance must hold across hospitals, populations, sample handling differences, disease stages, and confounding conditions. The model must not simply memorize one dataset. It must generalize. It must be interpretable enough for clinical trust. It must be tested prospectively. It must prove that it improves patient care. A promising blood test is not automatically a clinical test. This is the fourth lesson. In nano-biosensing, sensitivity is not enough. Specificity matters. Reproducibility matters. Clinical context matters. False positives matter. False negatives matter. Workflow matters. Cost matters. Regulation matters. And the consequence of the result matters. A test that tells a patient they may have a brain tumor cannot be casually wrong. A test that misses a tumor cannot be casually trusted. Liquid biopsy is powerful because it is less invasive. But less invasive does not mean lower responsibility. A blood test can change a life. It can trigger imaging, anxiety, treatment, or reassurance, so the standard must be high. Still, the convergence is extraordinary. On one side, micro and nanorobots may carry therapy into the body with greater precision. On the other side, biosensors may read disease signals from blood with greater sensitivity. One technology moves toward disease. The other listens for disease. One acts, one senses. Together, they point toward a new kind of medicine. A medicine of tiny agents and large interpretation. A medicine where the smallest engineered systems carry the heaviest clinical meaning. This is not yet the medicine of swarms repairing every cell. It is not yet the science fiction future, but it is very close in spirit. Because the core idea has arrived. Medicine does not have to remain macroscopic. It does not have to act only at the scale of organs, pills, scans, and surgeries. It can act at the scale where disease begins, the microenvironment, the vessel, the tumor surface, the infected airway, the molecular signal, the protein corona, the nanoscale sensor response, the hidden pattern in blood. That is the deep change. The battlefield is shrinking. But the meaning is growing. And as the battlefield shrinks, the ethical questions grow sharper. What materials are safe inside the body? How long should they stay? Who monitors long-term effects? Can they cross into organs where they are not wanted? Can they accumulate? Can they trigger immune reactions? Can they be manufactured consistently? Can they be tracked in real time? Can they be retrieved or degraded? Can the public trust them? How do we prevent hype clinics from selling unapproved nanoscale miracles? How do regulators evaluate something that is part drug, part device, part material, part imaging agent, and part robot? These are not side questions. They are the road, because the future of medical micro and nanorobotics will be decided not only by invention, but by trust. Trust in safety, trust in evidence, trust in manufacturing, Trust in imaging. Trust in algorithms. Trust in clinical benefit. Trust that a tiny device placed inside the body will not become a large problem later. The FDA and other regulators already recognize that nanomaterials may behave differently from conventional materials because size can change physical, chemical, and biological properties. That is exactly why nanoscale medicine is powerful, and exactly why it needs scrutiny. At small scales, surface area matters, shape matters, charge matters, coating matters, protein binding matters, aggregation matters, clearance matters. The body sees not only what something is made of, but how it presents itself. A carbon nanotube wrapped with DNA behaves differently from a bulk piece of carbon. A hydrogel microbubble behaves differently from a conventional drug tablet. A magnetic nanoparticle inside a microrobot behaves differently from iron in a tool. Scale changes behavior. That is the promise and the risk. So what are medical micro and nanorobots truly about? They are about tiny machines. But tiny machines are only the surface. They are about direction, about turning therapy from a flood into a guided act. They are about reaching places where ordinary drugs are too diffuse, too toxic, or too weak. They are about lung treatment that may someday move beyond passive inhalation. They are about bubbles that can move, signal, and release. They are about materials that can enter the body, perform a task, and disappear safely. They are about liquid biopsies that may detect disease without cutting into the patient. They are about carbon nanotube, nanosensors that translate molecular binding into optical patterns. They are about machine learning that sees disease signatures in signals too complex for ordinary interpretation. And they are about the possibility that future medicine will become both smaller and more exact. But the smaller the tool, the greater the need for discipline. The body is not a playground for clever machines. It is a living system, a person, a patient, a life. Every tiny robot, every nanosensor, every diagnostic algorithm, every engineered particle must answer the same question. Do you help enough to justify entering the body? That is the moral center of the field. Not novelty, not spectacle, not the thrill of saying nanobot. Benefit, safety, precision, evidence, trust. If this field succeeds, the future may look very different. A patient with a suspected brain tumor might one day receive a blood test that reads a nanosensor fingerprint and helps guide imaging or diagnosis. A patient with a localized tumor might receive robotic carriers that concentrate therapy where it is needed. A patient with a lung infection might inhale active drug carriers designed to remain in the lungs long enough to fight disease directly. A clinician might watch tiny therapeutic systems move through imaging, guide them, trigger them, and know when they have done their work. This is not tomorrow's routine medicine, but it is no longer pure fantasy. The pieces are emerging: movement, guidance, cargo, imaging, biodegradability, machine perception, liquid biopsy, preclinical efficacy. The next challenge is integration into real care. That is always the hard part. Science can open the door. Medicine must walk through it carefully. I'm Ralph Clayton, host of the VTM Podcast. Thank you for joining me for episode 23, and this look into medical micro and nanorobots, nano-biosensors, liquid biopsy, and the tiny technologies beginning to reshape precision medicine. And remember, not every nanobot is science fiction. Not every tiny machine belongs inside the body. And sometimes, the future of medicine begins when healing learns to move at the same scale as disease.